All right, let's continue. So in the yesterday class we started discussing about the enzymology of the DNA replication, right? And we studied that of course every single enzyme is required for the process of the DNA replication, and we understood that among all the most important enzyme is what? DNA polymerase enzyme. I believe you all remember in case of a prokaryotic organism what we studied, we have a 3 type of a DNA polymerases. When it comes to the eukaryotic organisms, we are having the 5 DNA polymerases. Slowly think and answer my question very correctly. My question is, in the prokaryotes, what is that enzyme that do not have a great role in the DNA repair mechanisms? Polymerase 3. Similarly, which is the homologous in eukaryotes? Which do not involved, I am saying delta. Okay. The delta is only responsible for what purpose? The accurate elongation purpose, right? I hope you all people remember, right? And what is that polymerase enzyme in prokaryotes having the ability to do the proofread even in the 5' to 3' direction? Which polymerase? Polymerase 1, which can do the proofreading in both directions, right? I hope you all people majorly remembered the 10 subunits of the polymerase 3 which I told you. It's very important, you all people has to remember that particular 10 subunits. Remember what are the major 10 subunits? Any one of you remember it? What are those? So it contain a core. What is that core? Is actually made up of alpha, epsilon, theta. Core connected to tau dimer, correct na? Then what do we called? Pol 3 prime, correct na? Then core tau connected to gamma complex, we called Pol 3 star. When this entire complex associated with the beta clamp, what do we call it as? Polymerase 3 holo enzyme. Holo represents generally complete enzyme, correct na? No point of getting confused over there. So I hope all of you understand here. In case of a prokaryotic organism, we all clearly understood and we all clearly studied that in case of prokaryotes, who is going to add the primer? What's that enzyme? RNA primase is the enzyme which generally adds the primer. But in case of a eukaryotic organism, we do not have any RNA primase. Which enzyme? DNA polymerase alpha add the primer, and what I told you, upon addition of the primer it is only start adding the nucleotides also, but it cannot add the nucleotides continuously because of its low processability. Low processability and no proofreading, that's the reason we cannot call it as the principal enzyme. And if you look into the polymerase beta, it never ever involved in the replication. Its ultimate function is what? Only repair. No point of getting confused over there. So the most principal enzyme in case of a eukaryotic DNA replication is what? Obviously polymerase delta, correct na? No point of confusions at all. What's the point you all people has to understand here? Polymerase delta major functions. It can do the proofreading, and why we are saying it as the most important enzyme among all, what will be the reason? Obviously the reason will be the highest processability. Now the question is super clear, everyone please continue. Why it is highest, why it is having highest processability? Because it is associated with a protein called as pCNA. So in case of prokaryotes, we generally call it as what? Beta clamp. Now you only tell me Kavana, how the beta clamp can be loaded on the core enzyme with the help of gamma complex. What do we call it as? Clamp loader. So similarly, if I want to place the pCNA onto the core enzyme also, I require clamp loader. So we don't have that. So here clamp loader having the another name we will discuss here. Then the next enzyme, DNA polymerase epsilon. I told you, right, we say that it may be a substitute for the DNA polymerase delta. Sometimes, in some books it was written very differently. In some books it says that it acts as a substituent for the polymerase delta in leading strand synthesis. In some books like Watson, it was written that it may act like a substitute for the polymerase delta in the lagging strand synthesis. So you remember, function is unclear, but remember as a substituent enzyme. The major point or the major difference between the polymerase delta and epsilon is polymerase delta having the proofreading activity, polymerase epsilon also having the proofreading activity. Polymerase delta will become highly processive only whenever it is associated with pCNA. But the speciality of the polymerase epsilon is that even though it is not associated, still it will be highly processive. That's an important point. So which of the following enzyme do not require the pCNA also, they will ask in the exam. To show the highest processivity, which is the polymerase epsilon. No point of getting confused over there. Everyone please continue, whomever joined right now. Now, what is that function of the pCNA that we studied? pCNA is generally called as proliferating cell nuclear antigen. Whatever the function performed by the beta sliding clamp, the same function performed by the pCNA. In case of a prokaryotes, to load the beta sliding clamp, we have the clamp loader, which we call it as gamma complex. In the eukaryotes, we call it as RFC, replication factor C. This is the one which is generally involved in the loading of the pCNA onto this. We can also say that RFC is the one which is responsible for the polymerase switching. What does it mean? So first the alpha will start it, then alpha replaced by delta. If delta wants to come into the picture, what is required? pCNA. If the pCNA wants to assemble, we require RFC. So alpha can be switched to the delta by this pCNA and RFC complex. I hope you remember. Anyway, we will see that. I hope you remember SSBP, single stranded binding proteins. What are their functions? They will be helpful in making the single stranded DNAs to become stable. Correct na? No point of confusion over there. Now the point is very, very clear that SSBPs in case of eukaryotes, we call it as RPA, replication protein A. No point of getting confused over there. Anyway, I will tell you everything in the process when we are reading it elaboratively, but for time being also, I want every one of you to remember origin. It never starts randomly. Correct na? So someone has to recognize the origin. So most of the people will think that helicase recognize the origin and it opens the strand. That is not possible. Someone has to recognize it, someone has to guide the helicase, then it will come and open. Now the enzyme that recognize the origin, we generally call it as an initiator protein. What do we call it as? Initiator protein. Never get confused. That initiator protein we generally call. What enough video? Okay. One second. All right. That is what exactly we call it as what? DNA A protein. DNA A protein is also called as what protein? Initiator protein. No point of getting confused. Helicase. In case of a prokaryotic organism, we call it as DNA B. In case of eukaryotic organism, we call it as MCM. Always remember the short forms. Okay. Helicase is called as what? MCM. In case of a prokaryotic organism, as I told you, helicase is the most important enzyme. Good. Helicase is the most important enzyme which open the double stranded DNA. We are going to read it at a higher speed. If this particular DNA B, that is helicase, wants to move with a higher speed, it always require an assistance protein, which is called as DNA C protein. Sometimes we call it as ATPase protein, which is providing enough energy for the DNA B to start opening this particular double stranded DNA. no point of confusing. Now the DnaB in case of prokaryotes, all of you remember, we call it as hexameric helicase. How many units are there? Only 6 units are there. The MCM in case of eukaryotes, that is only important for the exam, having 7 to 10 subunits. No point of confusing at all. And you know what is the DNA polymerase enzyme, and what is the enzyme that add the primer, we can call it RNA primase. That RNA primase in case of a prokaryotic organism, we call it as DnaG. It is very important, you have to remember the other names compulsory. SSB, Topoisomerase, which are the most common enzymes. Alright. Now, so far whatever we have studied is only one enzyme we studied. That is the major principal enzyme, that is the polymerase enzyme. Of course, there are many other enzymes are also involved. Knowing every single enzyme is important. See, sometimes in the last 2 papers you got so simple questions from the enzymes, 2 marks are in our pocket easily. Now, primase is also called as DnaG in E. coli. In prokaryotes, sorry, in eukaryotes, DNA polymerase alpha. Correct na? In case of a eukaryotes. What's the function of a primase? We said, right? Primase is the one which is adding primer. Correct na? So by that polymerase will come and elongate it. Now question is super clear. This particular primase, how many number of nucleotides that the primer is composed of? The average length in vivo, in the body, it is 11 plus or minus 1. That's all. Very small primer it is. In case of a laboratory or in vitro, it is up to 60 nucleotides. Now, every one of you remember, in case of a prokaryotic organism, the primer generally rich of this particular sequence, which is the GTA sequence. I want all of you to remember this particular statements compulsory for the exam. You can see the picture very clearly here. What's happening? This will be the DNA template. One second. Okay. So DNA template associated with this particular primer, then immediately polymerase enzyme will come and elongate it. Right. Anyway, I'll tell you in a simple words. What is the function of DNA? I'll explain you elaboratively in the process. DNA is called as initiator protein. DNA B is called as helicase. DNA C is the assistant protein, right? And what is topoisomerase? Correct na? Till here we call it as pre-primosome complex. In case of prokaryotes, pre-primosome. When I add the primer, what happens? Pre-primosome converted to the primosome complex. So whenever the primosome complex got formed, which indicates that initiation is completed. Then because the polymerase is coming and doing it, so polymerase only do the elongation process. Polymerase do not have any role in the initiation. Initiation, what is there? We have to add the primer, then that can be continuously elongated by this particular DNA polymerase enzyme. Okay, remember the difference between the pre-primosome as well as the primosome complex. Now, this is the points most of the students tend to skip it. These only you people has to focus, because in the given exam they always raise the question about the structure of the enzymes, which most of the students neglect. They just read 1 or 2 functions of the enzyme and we will leave it. Now, DnaG having, see, let's consider this is a protein, which is the DnaG. Every protein having what? N-terminal domain, C-terminal domain, then it is also having a central domain. Got it? N-terminal, C-terminal and central domain. No point of getting confused over there. And what is the function of a DnaG? Adding the primer. Correct na? Where it will add the primer? This is 2 single strands of the DNA. Correct or not? So remember, N-terminal domain, all of you focus, it is a zinc binding domain, which tetrahedrally ligands zinc ion via 3 cysteine residues and a histidine residues and implicated in recognizing single strand DNA. This is the exact question that you people can expect in the exam. Remember, the N-terminal domain of this particular enzyme contain 2 amino acids. Cysteine also it contain, histidine is also it contain. These two can be connected via zinc. This is called as N-terminal domain. What's the function of N-terminal domain? First, if someone recognize the single strand, then only they can add the primer. How they recognize? They recognize with the help of N-terminal. So listen again, only recognition can be mediated by this. Correct now? But what is ultimate goal? Synthesizing the primer. So that is mediated by a central domain. Why? Central domain is the one acting like a RNA polymerase. This is the direct question that we can expect. Which of the following domain of the DnaG having a polymerase activity? That's an important statement. Which is the central domain. That is the one which will carry out the protein synthesis. Now, if everyone quickly recall, this particular C domain, it is always associated with the DnaB. What is DnaB? Helicase, right? So, a C-terminal helicase binding domain, that also a statement in the exam. So, N-terminal domain is only responsible for recognizing the single stranded DNAs. Central domain we can call RNA polymerase domain, whereas C-terminal domain, what do we can call it as helicase binding domain. No point of getting confused over there, everyone remember it, compulsory for the exam. Now, what is this enzyme? Helicase. DnaB. In case of eukaryotes, we will read over there, we call it as what? MCM. Now, look into this, what you people can look at, observe here, helicase is actually 6 units, which is a hexameric helicase. It look like a wheel. I'll show you in the animations. It's keep moving, keep rolling, keep rolling, keep rolling, and keep moving, and keep unzipping the hydrogen bonds or breaking the hydrogen bonds on a continuous manner. No point of confusing over there. So now question is very clear. From where they start breaking the hydrogen bonds? From the origin. Now, how do they know that where is the origin? So someone has to guide them. And moreover, if the DnaB wants to open the hydrogen bond, it's compulsory require the energy. So if the DnaB wants to work, it require compulsory the assistance of DNA A and C. DNA A helpful in the recognition, DNA C is assisting in terms of the opening. You are getting the point. Once the DnaB placed over there, sometimes DNA C will be present there itself, sometimes it will get dissociated. So majorly I can say that DNA C is helping DnaB until it starts the process of opening the hydrogen bonds. Sometimes it may be there or it may fall down also. Now, it hydrolyzes the ATP when bound to single stranded DNA and opens up a helix as it moves along the DNA. Of course, in single second it breaks 1000 base pair. This is one of the reason it is strictly ATP dependent, right? One of the major enzyme that is ATP dependent. How many domains are there in the primase? 3 domains, absolutely correct. Similarly, very important statement for the exam, okay. E. coli DnaB protein, which is a hexameric helicase of identical 471 residue subunits. What is their function? Opening the double stranded DNA. Very, very, very important for the exam. Helicase always moves in this direction, 5' to 3' direction. Another very crucial point, majorly it dependent on the ATP, very important for the exam. Sometimes apart from the ATP, it can also use GTP, it can also use CTP, but it never ever uses UTP. That is also a question. Which of the following energy precursors cannot be used? Cannot be used by this particular helicase. Now, just on the previous slide we read one point. What is the point that we read? DNA, what is the enzyme we read? Primase. Primase is connected to what? DnaB helicase, which with the help of what domain? C domain, which means that DnaB also having some domain that can connect it to the C terminal domain of the DnaG, correct or not? Suppose example, this is the DNA primase, N domain, central domain, C domain. Exactly to the C domain, DNA B will come and attaches. Correct or not? So now all of you look into the picture. No point of getting confused. How this particular helicase looks like, it's a little different, which is a hexagonal structure. Obviously it is a protein having N-terminal domain and having C-terminal domain. Now tell me, C-terminal domain of the helicase will show that helicase function. You are getting my point? And what is the function of N-terminal domain? This is the one which is connected to the C-terminal domain of primase. You are getting? So again they will give the opposite statement in the exam, because we tend to forget it very faster. So this is the primase, N-terminal and C-terminal. No point of getting confused. Suppose example, it is this enzyme, what we can call the helicase, right? This is the linkase, right? This will be the linkase. C-terminal domain of the primase is connected to the N-terminal domain of helicase. That's a very important statement you have to remember for the exam. In the exam, they will give the opposite statement. Mostly people won't read only this. They just focus on the functions and they will just leave it. Okay. We can't predict the questions in the mol bio. Next, another very important and unique enzyme that we are studying is the, what is that? SSBP. What is the speciality of this particular SSBPs? Single stranded binding proteins. What is the speciality of this particular SSBPs? Now listen, I already told you, these two are the two single stranded DNAs, both got separated. Now what happens? They will go and attaches to them, right? Why? What is the major function? Number one is preventing the again double stranded DNA. In simple words, preventing the re-annealing. We also call it as rejoining. You got the point correctly? Now what else? I told you, whenever the single strands are opened, there is a great possibility that the single stranded DNAs undergo the coiling. Hairpin like structures they may form. So to prevent coiling or to prevent hairpin like structures, that is also mediated by the SSB, right? At the same time, remember all the time my words, when the double stranded DNA is in a double stranded form, most stable. When the DNA separates into two single strands, they are more prone to degraded by the vulnerable enzymes such as exonucleases, endonucleases. They keep on moving in the cytoplasm and nucleus. If they find something is unprotected, they will come and degrade it. So again, but if the SSBPs are attached to this particular single strand, enzymes cannot degrade them. So they will prevent the reannealing, they protect the degradation by the nucleases. At the same time, they prevent the hairpin loop like structures also, right? These are the major functions performed by this particular SSB proteins. Now you all people observe the picture so clearly. Sorry. Yeah, if you observe it, how many SSBPs will actually comes and attaches? This is the most important statement to be remembered. This is one SSB. One SSBP protein will provide a greater binding site to the another SSBP protein. Then second one will provide the binding site for third one will provide the binding site for fourth one, and so on. In this way, all more than 60 SSBPs will come and attaches to the single stranded DNA. So again, the question that they will raise in the exam is, all the SSBPs bind to the single stranded DNA in a non-cooperative fashion. Is this true or false? Obviously it's a false statement, because one is providing the binding site to the other, hence we can say that all are binding in a cooperative fashion only. Okay, that statement seems to be simple, but if you don't know it 200% you will make it wrong. Similarly, again let me recap it, no point of getting confused over there. I'll recap it here. DnaG, how many domains? DnaG, 3 domains, N-terminal, central domain and C-terminal. Correct, na? Function of N-terminal? Very good. Recognition. Function of central terminal? RNA polymerase activity. Function of C-terminal domain? Binding to the helicase. That's a very important statement to be understood. What is the next enzyme? Helicase, having only 2 domains. It's a hexagonal, but having 2 domains, N-terminal, C-terminal. Which is performing the helicase activity? C-terminal domain perform the helicase activity. N-terminal domain attaches to the C-terminal domain of the primase. Clear? No confusions at all. Now, what about the SSB? It's very simple. Okay. So, listen. E. coli SSB is a homotetramer. That's an important statement. SSB binds to the single-stranded DNA in several distinct modes referred to as SSBN. N represents number. So, all are not going at a time. One will attach us, one provide the binding site to the another. Like that, minimum of 35 to 65 SSBs will be associated. Only SSB having N-terminal as well as the C-terminal domain. In very simple words, SSB attaches to the DNA via N-terminal. One SSB provide the binding site to the another SSB at the, you are getting my point correctly. So, it attaches via to the DNA via N-terminal, provide the binding site to the next SSB via C-terminal domain. No point of getting confused over there, don't get confused. It is a very generalized information, but it is a more informative, never ever skip it. If you skip it, you lose the mark. Alright, so the next, another versatile enzyme, that is what exactly we can call topoisomerase. Multiple times the question was asked from the topoisomerase enzyme also, we have to know about it. Sometimes in the prokaryotes, instead of topoisomerase enzyme, they use the word called as Gyrase. Right. Now, what exactly the function of the topoisomerase? How fast the helicase is moving? What we said, helicase is moving how much fast? Helicase is moving 1000 base pair in one single second. So, suppose example, if you consider this is a thread, right? So, on one hand we hold the thread, on another hand by using thumb we keep opening the thread. So, what is happening over there? The thumb or the hand creating a pressure on this particular thread. Because of the pressure, what happens? The thread undergo the coiling. Whenever my fingers reach this area, how can I separate? I can't separate further. What is the solution? I have to remove it. Sometimes removing this coiling is not that much easy. That time I will cut it. You are getting the point. I will cut it, I will join it, then again I will start continuing the function. You are getting the point. Similarly, so instead of the fingers, you just replace with the helicase. Whenever the helicase is moving at a higher speed, it creates a greater tension or stress on the DNA. So, as a result, DNA undergoes the coiling. So, when the helicase enzyme reached at this particular point, it cannot move further. That time, topoisomerase will come. So, this is a very stressful situation. Why? Because anyway, topoisomerase will come very faster. Because helicase will move very faster, no one knows that where exactly coiling will form. Correct or not? Coiling may form here also, here also, here also, anywhere it may form. That's why topoisomerase keep following the helicase. The moment or the area where the coiling occur, it starts removing it. How? What is the mechanism that is involved in removing? Every one of you please try to understand, see an average. Opening the double-stranded DNA by helicase will create a supercoil ahead of the replication fork. Correct na? Every 10 base pairs, such an important statement, replicated at the fork corresponds to one complete turn about the axis of the parental double helix. What does it mean? We all read in the biochemistry, in the nucleic acid will read it. Let's, what kind of a DNA present in our body? bDNA, correct na? This is what we can call as one turn, correct na? One turn of the bDNA contain 10.4 nucleotides, correct na? So we called it as one turn. Obviously we have to go many turns, go through many turns. Whenever the helicase starts moving one turn, mostly the coiling occur. That is what exactly they are saying. Every 10 base pairs replicated at the fork corresponds to what? One turn, because one turn contains that many number of nucleotides. So every turn there is a possibility that helicase can create that particular stress. So listen, therefore for a replication fork to move, the entire chromosome ahead of the fork would normally have to rotate rapidly, correct or not? If the coiling occurred here itself, how can I rotate? How can I move? So that is the reason helicase is immediately followed by the topoisomerase enzyme. Now that supercoiling or the stress or torsion or the tension should be released by an enzyme called as topoisomerases. Understand everyone very correctly? Okay. Now how can we remove this? This is the more important point to be understood for the exam, because the questions always comes from the mechanisms. Everyone please continue. See how it will doing. There are different type of topoisomerases are there. We can call type 1 and we can call type 2. In the type 1 we are having type 1A, we are having type 1B. Now some topoisomerase 1 only act on single strand, only act on single strand. Topoisomerase 2 act on the both strand. So what's the mechanism? Simple mechanisms. Consider this is the DNA. All of just use the brain. This is how the coiling occur. Let's consider exactly here coiling occur. Now tell me what should I do now? So I will first break it. Correct na? First I will create a nick. What do we call? Nick. So through that gap we will pass them and again we will seal that particular nick. We will see that. So first cleavage of one or both strands of DNA, then passage of one segment through that particular break, then sealing the nick. Let's try to understand more elaboratively how exactly it happens. So please try to remember maximum amount of the information here. Everyone please continue. How many types of the DNA topoisomerases are there? 3. 2, 1A, 1B and 2. I will explain you elaboratively, just follow me. Type 1A, 1B almost similar, because they are showing what mechanism? Acting on only one strand. Now let's understand. Type 2 will be working on both strands. I will show you in the next class also in the animation, but let's try to understand here overview. If you look into the type 1A topoisomerase, all the students look into this. This is the type 1A topoisomerase. First of all, what should I do? I have to cut. If I want to cut, what bond I have to cut? Hydrogen bond will cut. Phosphodiester bond. We are not separating, right? If you want to separate, we will do the hydrogen bond. We have to cut what bond? Phosphodiester bond. I hope you all remember. If I want to cut the phosphodiester bond, what kind of attack is possible? Nucleophilic attack, which means we require oxygen here. So in the topoisomerase enzyme, on the active site, tyrosine amino acid is present. Tyrosine amino acid, what is the side chain? OH group, correct or not? I hope everyone is getting. So where oxygen is acting as a nucleophile. No point of getting confused over there. That also they will raise the question in the exam. Now you can see that particular topoisomerase, see this, how many strands are there? 2 strands are there, green color strand and purple color strand. Now what's happening now? Now this particular this is the topoisomerase enzyme, scissor. What it is doing? It is having tyrosine having OH, oxygen having nucleophile, nucleophilically come and cut it. Correct na? The purple strand got cut. What happened when we cut it? There is a gap. Correct or not? Through that gap, we are passing the green strand. You are getting the point? Because gap was created, like this I can easily pass it, right? So once I pass it, it can easily release it out from that particular coiling. Coiling can be removed, then again helicase will continuously move, then it starts opening the hydrogen, breaking the hydrogen bonds. This is what a simple mechanism of the type 1A, which is having tyrosine in its active site, which will come and cut one strand. Through the gap created, it is passing the other strand. Simple mechanism, not complicated. Now, how can I differentiate type 1B with the type 1A? See, this is a very kind of a different one. Okay, see, it is also producing a transient single strand break. First point, only single strand, not the double stranded. This break in the phosphodiester backbone allows two sections of DNA helix on either side of the nick to rotate freely relative to each other. Remember that statement, rotate relatively free to each other. Now you people only observe this. This is the DNA which undergone the coiling. Now you can see that this is what enzyme? Type 1B DNA topoisomerase. What amino acid is there in the active site? Tyrosine amino acid. Now you can see the picture, tyrosine amino acid with the help of a hydroxyl group, it will come and create the break, what we can call nick, single stranded break. Now all of you focus. Now can I say one point to you? Now this strand and this area and this area got separated. Can I say that they are having a lot of stability? They won't be greatly stable. Can I say that I can rotate however I want? Correct or not? So now in a simple words I will tell you, in a very simple words, look into this. You can see here, suppose example, this is how my DNA look like. See the arrow, this is how it will turn actually. Because of the stress, it turned in this way. Now I can cut it and I can bring it back to the normal direction. Same thing will happen here. Now in order to remove that coil, first we will create the cut, then the two ends of the double stranded DNA now rotate. In very simple words, this is the original direction, it twisted like this because of the coiling, because of the stress. Now I am bringing back to the normal position just by rotating it. But if I want to rotate it, what I require? A break. So I have to, I have to make sure that it never be stable. I have to make sure that it will be flexible. So to make the flexibility only, we will create that particular nick. So the original phosphodiester bond energy is stored in the phosphotyrosine linkage, making the reaction reversible. Simple. We just twisted in such a way that later phosphodiester bond can be rejoined with the help of a ligase enzyme. No point of getting confused over there, and it is easy to understand. Sometimes in the exam they will give you a very crystal clear point. Listen my statements correctly. 1A, 1B, they will say that ligase is not required for the 1A and 1B. Sometimes this statement is true statement. Why? Because in very simple words, more specifically I can tell you, this is a nucleotide, everyone focus. These are the nitrogen bases and this is what? Phosphate group. Correct na? Now what I broke actually? Phosphate group only I broke. I broke the phosphate group in such a way that I can rotate it easily. Correct na? Sometimes after rotation, see, remember my words, I always use a word that I break the phosphate. I am never using the word that the phosphate is released out. You are getting my point? Phosphate is present there itself. Okay, I just break it to make it rotate relatively free to each other. Now when I am rotating back, can I say that again phosphate will be added there itself? You are getting the point? I just created a gap to rotate. As a result, once again the phosphodiester bond will be reformed. You are getting the point? Hence there is no requirement of ligase. You are getting the point? In case if the reformation is not happened, that time we will use the ligase. That's a very unique statement to be understood. You will understand in the exam. Spontaneous reformation of the phosphodiester bond regenerates both the DNA helix and the DNA topoisomerase. Very simple, no point of getting confused over there, everyone understand. So, next another unique enzyme is the topoisomerase 2, which is only acting on how many strands? Both strands. It will cut both strands at a time. Type 1A and 1B are specifically cleaving only one strand. It is named as what? Gyrase. And very important statement again, it is a what? A heterotetramer. It is having 2 units of A and 2 units of B. And it cuts phosphodiester bonds on both strands of the double stranded DNA. Now again, now you tell me in the topoisomerase 1A and 1B, have we used ATP anywhere? No. We used ligase anywhere? No. Mostly reformation occur. But here we require ligase, at the same time we compulsory require the ATP molecule. This is one of the statements that they can raise in the given question. Which enzyme is ATP dependent and which enzyme is ATP independent? This is then DNA. DNA is generally called as negatively supercoiled. DNA generally called as, this is called negatively supercoiling. This is called negatively supercoiling. I know, same thing will happen. 1 second. Okay. What happened in this particular negatively supercoiling? Now, because of the stress created, how does the negative supercoiling is looking like? You can see that it is a kind of right-handed conformation. Sorry, left-handed conformation. You can see this like this. Okay. Now, you can see this. This is what original, this is the original form. Now, because of that stress, now you can see that right hand it is. What do we can call it? It as what? Positive supercoil. Original form is the negative, but because of the stress it is converted into the positive supercoil. Now, what is happening? What should I do now? I have to cut both strands because the enzyme involved is the topoisomerase 2. Having how many subunits? 2 units of A and 2 units of B. You people only try to observe that 4 units coming close together. They are calling it as heterodimer or tetramer, you can call it. 2 dimers are there. Correct now? It will become the tetramer now. You see that what it is doing actually? It is cleaving one double stranded DNA. You are getting my point. In the picture it was not shown correctly. I will show you the animation. It cut what? Both strands. It cut the both strand through the both strand, whatever the gap created by through because of the breakage of both strands, it will pass another homologous DNA. In a simple example, this is the DNA, right? What we cut? We cut the both strands. Correct na? Through both strands, we are passing a homologous DNA just to remove this particular coiling. Otherwise, I will explain in this picture only. All of you look into this. First, all of you focus. 1 unit of A and B, another unit of the A and B first created the cut. Now, observe it. Everyone focus on the picture so clearly. What this particular A and B is doing? This unit of A and B pulling this chain. Can you see that? Pulling this chain to come backwards of it. You can observe here. Okay. This 1 unit of A and B pulling this particular DNA to come backward of this until entire stretch come out. You are getting my point? Until entire stretch come out. The moment entire stretch come out, anyway their ligase will come and seal that particular nick. Okay. I will show you in the next class with the help of animation. Just remember, this is how the cut happened. Okay. So, from here we will pull the strand, we are bringing it back, there we will seal it. We are pulling it first to the front, then front to the back, there we will seal it off. Hence, we can create this particular negative supercoil. In more simple words, remember the original supercoiling is like this, original supercoiling. But how it happened right now? This is how it formed. Correct na? Now you tell me, how can I bring this to this form? I have to reverse it. Correct or not? I have to reverse it. So from where I will start it? I will start from this particular point. So simple. I will start from this particular point. I can bring it here. In more simple words, this is the original form. Let's consider this is what? Original form, which is the reverse C. Okay. Because of the coiling, it became normal C. Now you tell me what I can do to bring it to the reverse C. From where I have to start? I will start from the downwards and I will go towards the upward direction. Correct na? That is what exactly happens here. You understand? So we are converting from a positive supercoil to the negative supercoil. For that reason, we are cleaving the both strands and we compulsory use the ATP and we compulsory require the ligase enzyme. In case of a 1A and 1B, in case of 1A and 1B, there is no requirement of the ligase. Okay. Now the last and final most important protein, this is also important for the exam. Yeah.
So the entire motto of this topoisomerase is to uncoil, right?
Uncoil, that's all.
Then why are we positive coiling, negative coiling?
Because the
Anyway.
that is true, but DNA is generally negatively supercoiled. See, I will explain you in the chemistry part. We have a DNA, bDNA, zDNA. This coiling occurs whenever the stress formed. Okay, how can we represent that particular stress? We can call it unwound, we can call it overwound. Okay, whenever suppose this is how the DNA look like. If the DNA undergo the coiling, how can we explain there is a coiling taken place? That we can explain by overwound or unwound, or overcoiling or the lower coiling, we can say that. So just that is a nomenclature that we can give to explain their tension. Okay, ultimate goal is of course to stretch it out and to release it out only, but how do we are stretching, in which direction we are moving? So to explain that, we use the word positive and negative supercoil. That's all. Understand correctly? That's all. It is just a nomenclature only. Now the last and final enzyme is the ligase, another most important enzyme. The moment you heard the name of a ligase enzyme, one of the common point you have to remember, ligase enzyme not only involved in the replication, for multiple functions ligase is required. We are going to read it. Splicing. What is splicing? Removal of the introns, addition of exon. Wherever we have to form the phosphodiester bond, there it will come. So replication, splicing, repair and recombination. In every function we require this particular ligase enzyme. Don't get confused. In what direction we form the phosphodiester bond? Phosphodiester bond formed between what? 3' hydroxyl group to the 5' phosphate group. Again important for the exam point of view. This ligase is also strictly energy dependent. But what kind of a energy precursor that it is using? It is strictly depends on organism. That's why you can see that free energy required by this reaction is obtained in a, this is the statement, in a species dependent manner. So in some organism it may be NAD, some organisms it may be ATP, some organisms it may be AM. So that's why we can call the species specific manner. Okay, but it is strictly energy dependent. There is no point of getting confused over there. All right. Now quickly answer these questions. Don't take too much of time. Everyone answer the questions. So of course, what is that enzyme? DNA polymerase 3 is the principal enzyme that is required for the process of the DNA replication. Quickly, all of you, DNA unwinding is done by helicase, correct na? There is a difference between the unwinding means unzipping, breaking the hydrogen bonds, whereas topoisomerase is required for removing the tension, torsion or the coiling. Correct, na? Which enzyme removes supercoiling in replicating DNA ahead of the replication fork is obviously topoisomerase enzyme. Simple they are. Next, all of you, what's that? B for ball. Yeah. Correct, now forgot Kavana, you have only 24 hours. Proofreading activity of DNA polymerase 3. Synthesis of a daughter strand always takes place in which direction? 5 prime to 3 prime, undoubtedly. Which means proofreading will obviously occur in 3 prime. I think you forgot about endonuclease and exonuclease, okay? Which is what activity? Exonuclease activity. No point of getting confused over there, right? Next, slowly. The reaction in DNA replication catalyzed by DNA ligase is C. Correct, na? So ligase is the one which is generally involved in formation of phosphodiester bond, and we know very well phosphodiester bond always formed between 3 prime hydroxyl group of one nucleotide to the 5 prime phosphate of the another nucleotide. There is no doubt about it. Answer will be the C. Right, clear everyone? Correct. Right. In the yesterday class and today class, what exactly we have studied is only a small part, okay, very small part we studied, okay. Now we are going to enter into the real process. Now we understand very clearly what are the rules for the DNA replication and what are the enzymes that are required for the DNA replication. Now let us understand what is the original process. How exactly, how accurately the process of the DNA replication will be taking place. Listen everyone, in any process, even it is a replication, transcription or translation, these 3 steps are common: initiation, elongation, then followed by the termination. So from now onwards lesson will become tougher. Once we go to the transcription, that is the most toughest part. Then imagine the translation that we don't understand only, okay. So listen this lesson carefully, okay. What is exactly the initiation? Tell me, you already understood. Tell me what's happening in the initiation. First there is a recognition of origin, then opening of the double strand, then while opening only tension will be created. Who is going to come now? Topoisomerase and SSBPs, followed by first only helicase, followed by primase. Polymerase never ever comes in the initiation. What is the use of polymerase? What will come? Primase. Primase will come and add the primer. Till addition of the primer is initiation. Correct or not? Recognizing the starting point, which is called ORC, separating the double stranded DNA by helicase. That time only obviously SSB will come, topoisomerase will come, then synthesis of primer. Later what is going to happen now? We are adding continuously DNTPs, correct na? We never add the NTPs, we only add the DNTPs. And what is the ultimate goal of the DNA polymerase? You know that? 2 functions. What is that? Polymerization. What is the meaning of polymerization? Formation of the phosphodiester bond. Next, proofreading, that's all. Proofreading it and formation of the phosphodiester bond. Once the entire DNA strand gets duplicated, what we gonna do? We are going to stop the replication, which we called termination. It seems to be easy, but it's extremely complicated. Now let's try to understand. Now tell me, Kavana, I hope you remember. In case of a prokaryotic organism, how many ORCs are present? Only 1, because prokaryotic organism itself is a small. In eukaryotes, we have more than 150 replicators are there. Anyway, let's understand. Let's consider, all of you understand my words correctly. Focus. This is what? DNA. Let's consider this is ORC. Now I am zooming or magnifying this ORC. How does it look like? One ORC only present, and it is made up of how many base pairs? 240 245 base pairs. Okay. Now I am writing the oriC like this. Okay. This is how many base pairs? 245 base pairs. This particular oriC arranged in the form of a boxes. Okay. How many boxes are there? All of you understand. 5 boxes are there. 1, 2, 3, 4, 5 boxes are there. Each box is made up of 9 base pairs. You are getting my point? Apart from this, again we are having on the left hand side 3 more boxes are there. How many? 3. Each box is made up of 13 base pairs. You are getting my point correctly. 245 base pair origin contain 5 boxes, each box is 9 base pair. On the left hand side 3 boxes are there, each one is how many? 13 base pair. Now I will ask you some important question. I want everyone use the brain and answer. Now first listen, this 9 base pair, it is the sequence TT, AT, CC, ACA. Is it necessary to remember? Remember it. Okay. Some sequences we have to remember this. And this sequence called as DNA A boxes. Now can anyone tell me why I am using the word DNA A boxes? One DNA? I hope you all remember, right? What is the function of DNA A? Initiator protein. Where exactly it recognizes oriC? Exactly at the 9 base pairs. That's the reason we called them as DNA A boxes. Now, how many here are there? 13 are there. We call it them as DUE. What does it mean? DNA unwinding elements. The area where the helicase starts unwinding. That's what we can call DNA unwinding. They are extremely made up of adenine and thymine. Now all of you understand my words correctly. Look into the picture, you get a good idea. What is this? You can see here, this is what? oriC. Correct na? See the DNA only very smaller, this is the oriC. oriC made up of 245 base pairs. On the right hand side 1, 2, 3, 4, 5 boxes are there called as DNA A boxes. Each box is of 9 base pair. On the left hand side 1, 2, 3 are there called as DUE. Each one is made up of and rich of adenine and thymine. Clear? No confusion at all. Okay. Now that is how the oriC is actually looking like. Now I am having a small doubt. Now my doubt is, all of you focus on the picture. This is the oriC, 9 base pairs, 13 base pairs. As you all said, who is going to recognize first? DNA A protein. This is called DNA A protein. Along with the DNA A protein, we require some more proteins called as HU, histone unit-like protein. Histone, it is not a histone, okay? Histone unit-like protein, which is also a type of initiator protein, along with the ATP molecule. Now, where they comes and attaches, can you see that? Exactly to which boxes? 9 base pair, which we called as DNA A boxes. Now, who has to come next after DNA A? Helicase, which is also called as DNA B, comes along with the function of C. This is very clear. Now, DNA B starts opening the double stranded DNA wherever the DNA A is present. Where the DNA A is associated? 9 base pair. But you see, opening is happening from 13 base pairs. What is the reason? Because the 13 base pair contain only adenine and thymine. Only 2 hydrogen bonds are there, then it is easy to break. Correct na? This entire complex only we call it as what? Pre-prime complex. No point of getting confused over there, easy to understand. Since what we are generally studying, E. coli. Correct na? E. coli DNA is what type of a DNA? Circular DNA. Correct? So, I already told to all of you very clearly, both prokaryotes and eukaryotes will show what kind of a replication? Bidirectional replication. If it is the origin, one fork is moving in one direction, another fork is moving in the opposite direction. Correct na? So, whenever the replication fork is moving continuously, the DNA of the E. coli look like Greek letter theta. That's the reason we call it, this replication is also called, this replication called as theta replication. You are getting the point? Don't get confused over there. Now, you only tell me what is the difference between the pre-primosome and the primosome complex. You can observe the picture very clearly here. Here primase is added, correct na? That's why we can call, if I remove this, it will become pre-primosome. Tell me what are all there here? A is there, B is there, B is associated with C. Sometimes C may fall. And what is this? Topoisomerase. So everyone use your brain. See, helicase is exactly followed by topoisomerase. Wherever coiling comes, it's keep removing. Otherwise it will stop. If it stops, process will stop. Correct na? Now, so whichever it is moving, which one? Helicase is moving. That is called as replication fork. That direction. That is the area where the replication will start. Now finally, primase will come and add the primer. Till here, what do we call it as? Active initiation complex. Initiation is over. It is so simple. Now what is most difficult? That is regulation. Okay. I'll explain in the next class, but I'll give you the overview here. What exactly the regulation? Now use your brain and answer the given question correctly. Now after the primase add the primer, which enzyme has to come? Polymerase has to come. It has to continuously synthesize the DNA, which is called as elongation. After elongation, termination. Now can I say one word? Before completing the elongation and termination, second round of initiation should not start. Does it make sense or not? Before completing the elongation and the termination of the first round of the DNA replication, the initiation of the second round should not start. You are getting the point correctly. See, you are starting a process. The process contains 3 steps: initiation, elongation, termination. Initiation is over. Next, and termination. Whenever the elongation is happening, there is a possibility that initiation occur again. Why? Why initiation occur again? The reason is that you can see here all the enzymes are free, right? Every enzyme is free. Now every enzyme can show their function, then again new round of initiation may start. Now inhibiting every enzyme activity is a complex process, correct or not? Instead of that, if I just block the oriC, entire enzymes will become inactive. I hope everyone is getting my point correctly. Instead of blocking the entire enzymes activity, we are just blocking the oriC, right? So this process we call it as sequestration. What is that? Okay, no point of getting confused. So easy. First time only we feel confused. Okay, so understand all of you. Post initiation oriC. What is the meaning of post initiation? Initiation is completed. Now this oriC is prevented from interacting with replication initiation machinery, because you can't block all enzymes. Instead of that, you can only block oriC. That mechanism only called as sequestration. You got it? It's not that much easy. We will understand in the next class how exactly this mechanism can be mediated. If you get a tough question, you will get from here only. Okay, no one will give just initiation, elongation and all. Okay, clear everyone, online people? Anyone having any doubts, you can ask. Okay, all right. Okay, tomorrow also same time. Okay, what we do is, okay, tomorrow also same 9:30 to 10:30 and 10:45 to 11:45. Tomorrow is Friday, right? Yes. Okay. Saturday we are having paper 1 from 9:30 to 12:30 with break. Okay. This is on Saturday, and Sunday I will take a full class. Okay. From 9:30 to 5 p.m. Okay. With lunch breaks and all. Okay. Clear everyone? Now we have to work very hard, for the same exam somewhere someone in India working 10 times harder than all of you. Okay, 10 times. So the more hard work we do, more easily we can crack. Clear everyone? All right, all right. That's all for now. I'll see you in the next class. Okay, stop it. We are rejoining, no? Information won't last, dear. Why it will last? Because we are cleaving what bond? What bond?